genomic epidemiological studies for public health applications. And I'll go ahead and turn it over to you, Allie.
Awesome, thank you. I will just play around with sharing my screen and okay. I'm hoping that folks can see that. I'm assuming so. It looks like you can. Okay, perfect. Yeah, well, thank you so much for having me. I love talking about genomic epi, and I think it's always exciting to get to think about these types of questions and to share them with other folks and get them thinking about these types of questions as well. So yeah, really appreciate being here for this seminar. And, I want to say that for all the rigor and methodology that surrounds traditional epidemiologic study design, right, that I think so many of us learn in the first courses that we take as part of an MPH or PhD program in epi, for all of that sort of canon that exists for surveillance epi or traditional epi, that really doesn't exist in the same way for genomic epi. And so I think a lot of this thinking about sampling design is often very pragmatic and practical, and is often based on experience and intuition, and in many ways sort of common sense thinking about factors at play. So oftentimes there is not a lot of perfect answers, and there are a lot of trade-offs. And I think that makes it both sometimes a little bit of a challenging area to engage in, but also a really rich area to think about. And so it's my pleasure to talk about it. And what I want to do is sort of provide sort of a summary initially, probably first 20 minutes or so, of how I try and approach sampling design for genomic epi studies in public health. And then I want to walk through an outbreak that occurred actually while I was a graduate student, and that I, as an academic genomic epi, collaborated with the Washington State Department of Health on. And I'm hoping that we can start to think about sampling design and genomic epi study design together with that as a work example. I personally feel like seminars are a little bit more fun when you get to engage with them. And so, yeah, there's going to be a little bit of request for interactivity towards the end of this talk. But before we dive into that, I'll start with some of the theory and the ways that I consider sampling and study design. So I really think that for all study design, regardless of whether this is epi or anything else, really, if you are thinking about research generally, applied, academic, otherwise, really it is useful to think first and foremost about what you're trying to do as the starting point for how you do study design. And I didn't make this up. I think this is like a fundamental principle that they'll often teach in graduate school. But I think one of the diagrams that I saw when I was a graduate student that best encapsulated this dynamic for me was from this professor, Lance Waller. He was I think it still is a spatial statistician, but he had this diagram that he called the whirling vortex. And basically in this whirling vortex, you have this never-ending cycle. But where you start is up here in the upper corner with the question that you want to answer. From there, you think about the data and the methods that you would need in order to answer the question that you are interested in. From there, you sort of compare that to the data that you can actually get access to and the methods that are actually available for your use. And that tells you something about then the question that you can answer, practically speaking, given the data and methods that are available to you. And again, you kind of compare that back to the question that you're actually interested in. And you're hoping here that the question that you can answer is actually a fairly good approximation of the question that you want to answer. And this is a whirling vortex because this is an iterative process. And when these, the question you can answer and the question you want to answer really aren't particularly aligned, that's when you start to think a little bit more about how to get better data, how to develop better methods, et cetera, et cetera. Interestingly, with genomic epidemiology, I think that, even though this is probably where we want to start with any type of study, the vast majority of genomic epidemiology has really started from this third box down here, data we can get and the methods that we can use. And I think, that's not ideally where we would start, but I would say that as a field, genomic epidemiology has often been built, and the canonical papers are really sort of starting from the data that we can get from convenient samples, from the methods that are available. And really what has changed this, I think, is the scale of data that have been generated during the pandemic. We've seen sequencing intensity and just absolute numbers of genomes at a scale that is just, completely out of proportion to anything that we've ever had before for pathogen genomics. And I think that that has opened up this incredible opportunity to really think a little bit more upstream. But what is important to know is that, we've often started here and we've learned a lot from genomic epidemiologic studies that do start here with the data that we can get. So don't let don't let perfect be the enemy of good. There's plenty that you can do from convenient samples. And we'll talk about this a little bit more, but that shouldn't prevent you from thinking about, in an idealized scenario, how you'd like this to go. All right, so I think that the nicest way to really think about this is to start from an actual question that you might want to investigate. So theoretically, this is the question that we want to answer. So as epidemiologists, we've detected an elevated incidence of a particular disease in a congregate living setting. That's not an uncommon situation to encounter. And the questions that we might be interested in are those cases related? Are they epidemiologically linked? And are we observing transmission that is occurring within this particular congregate living setting or skilled nursing facility? Or do we have some sort of active surveillance program going on and we're simply finding unrelated prevalent cases or distinct introductions into this facility of lineages or infections that were acquired elsewhere in the community, but just because really we have better surveillance within this setting, we're capturing those community-acquired infections more readily. So this is the question that we want to answer. And so from there, we go into what data can we get and what methods can we use. So the first thing that I try to think about is which specimens do I want to try and sequence off of, and what types of specimens are actually available to me, what data are available to me. And this is often shaped by pragmatism and by trade-offs. And I think that when you're thinking about which samples you want to source as the samples that you'll sequence off of to generate this data for your study, I think about this really as three bins of criteria for guiding this. You can think about the type of specimen that you want to try and access. You want to think about how to optimize the biological quality of that specimen in order to get really nice, high quality genomic sequence data. And then, because this is such a backbone of genomic epidemiology, you want to think about what metadata or epidemiologic data or clinical data are associated with that sample and available to you to use as well. So for example here, if I'm thinking about what type of specimen is available to me, often this is, say, a direct diagnostic specimen, nasal swab, or, serum from a blood draw, a CSF sample, something like that. And The other sort of like, so you have diagnostic specimens, but the other specimen type that's often available to you is a cultured isolate. We see this a lot for bacterial diseases that clinical microbiology labs will often culture an isolate and send that to our public health lab, and that'll be the starting specimen that'll be available to us for sequencing. I think often type of specimen and biological quality of the specimen are sometimes in they balance each other out, I think often. It's hard to get both to be really optimized, although you're often trying to do that. So when you're working off of a diagnostic specimen, what can be really nice about that is that you are often able to sequence off of those types of specimens in a faster turnaround time. And so if you are really trying to do this study quickly to get inference so that you can bring this outbreak under control, maybe it is worth it to you to have a harder specimen to sequence off of, but that you can get those data more quickly for. A cultured isolate often has a higher sort of biological quality from a sequence data perspective because there is more pathogen available in the sample. And really, genomic quality that you get is highly correlated with the amount of pathogen that's actually in the sample. And so a cultured isolate will often give you nicer data, but you might have to wait longer for that pathogen to grow up and develop that culture. And especially for pathogens that are slow growing, that might be a longer time period than you really want to wait. If you are working off of diagnostic specimens, I think some of the best things that you can do is really think about how to capture a high quality specimen and what factors shape viral load in the infected individual. And you want to look at the literature oftentimes to see what those shedding dynamics look like. How long after symptom onset really do you have to get that diagnostic specimen to capture a reasonable amount of pathogen load in that specimen that could be useful for sequencing? So a lot of literature searching, a lot of thinking about what you want to do here. And then this third piece, metadata associated with the sample, I think that can, that can sometimes be overlooked. It's really, really important. So even if you have all the sequence data in the world, typically your ability to do genomic epidemiologic inference will be pretty hampered if you don't have information about when those specimens were collected from the infected individual, information that you might want to associate with the sequence data, exposures, like all of that rich epidemiologic data that we would typically use. Really, those data are critically important to understanding the dynamics that we see in the genomic sequence data. And so even if you can get a really high quality specimen really quickly, if it has no associated metadata, that's probably not going to be super helpful. And so really you're trying to balance all of this when you're trying to think about which specimens or which samples you're actually going to collect. From there, you might be thinking, okay, well, how many samples do I want to collect in order to investigate this question of interest, where we're trying to understand relationships between these cases in this congregate living facility? And I will say that this is just like a super underdeveloped area of genomic epidemiology. We have very clear ways to do sample size calculations in traditional epi. I would say in my experience as an academic, many groups, if they had one study or one question, would try and answer this with simulation-based inference. So they would model the transmission dynamics or the outbreak dynamics that they were hoping to learn about in their genomic epi study. And they would run some simulations to understand how many samples they would need to get in order to get good visibility into those transmission dynamics. That approach, is technical and is also particularly well suited when you have a single study that might take up like your next 2 years. it's not as well suited to the frequency with which we're investigating these different questions or having this sort of ongoing need to do this work. And so that has led, I'd say, like really Shirley Wool and other folks, but I think really her, she's done the majority of this theoretical development to try and make more of this sampling size calculation theory for genomic epidemiology. And there's really kind of two approaches to this. There's looking at number of samples that you need to correctly capture true direct linkages between pairs of cases. And then, and then there's also this sort of broader surveillance picture of how many samples do I need in order to accurately measure variant prevalence, or in order to know when I have a variant that is circulating in my jurisdiction at a particular frequency. So these are the areas where theory actually exists. I would love to see more theory exist, but oftentimes how many samples you're trying to include to answer your question is a matter of intuition right now, which I agree isn't ideal, but that is sort of the reality of it. I think the other thing that is important to flag is that how many samples you need depends, at least partly, if not largely, on the actual epidemiologic dynamics of the outbreak at play. And this is hard to know ahead of time, right? Where if the whole point of this study is to understand the epidemiology of the outbreak, then it's hard to know how many samples you need in order to understand that epidemiology effectively. You can do this sort of review loop after the fact, though, and I think there's an example of this here. So in this study, this was looking at Zika virus introductions and endemic spread in Colombia. And we had very, very little sequence data available for looking at this study. And the question became, had we sampled enough genomic diversity from Colombia in order to accurately capture the true number of introductions that had seeded the larger outbreak within Colombia? And one of the ways that we looked at this was basically to do these after the fact rarefaction curves that look at the numbers of introductions that you would observe when you down sample the data and include even less. And the goal here is to try and understand how the number of introductions that you observe is changing as a function of the amount of sequence data that you include in your analysis. And so here I'm comparing two countries that had very different outbreaks that occurred at very different times in the sort of America's-wide epidemic of Zika virus. And you can see here that this rarefaction curve for looking at number of introductions into Colombia as a function of the sampling intensity from Zika viruses in Colombia basically plateaus by the time we get to 10 sequences. So even as we add more genomic data sampled from Colombia, we don't actually see that partitioning into further and further numbers of introductions into the country. And that contrasts with the dynamic that we see in Mexico, whereby this curve really isn't plateauing at all at the max number of sequences that were available at the time that I did this analysis. And this evidence is really showing that the more that we sequence Zika viruses from cases from Mexico, that really the more introductions of Zika virus we start to see contributing to the broader epidemic in Mexico. And the fact that this hasn't plateaued means that probably if we were to sequence more data from Mexico, this would start to increase. And so this is an example that the number of samples that we need to understand the epidemiologic dynamics in these different countries is tied to the actual epidemiology of Zika virus in these countries. The Zika virus epidemic that occurred in Colombia happened quite early on after the introduction to Brazil, when many other countries weren't as heavily affected. And so just the force of infection and the numbers of countries that could seed or sort of donate introductions to the country where it's limited. And that looks very different in Mexico, where this outbreak hit a little bit later on, once there were a lot of different countries that were concurrently experiencing Zika epidemics. And another thing to think about is really, how are these samples collected? And this is something that we think about in EPI all the time, right? Like, what is your sampling frame? I tend to bin this into really 3 sampling frames that are available to you. You can have sequence data that are representatively sampled. You can have sequence data that are sampled through a convenience sample. And then you can have an intensive sample where you're really trying to sequence as many cases as possible from a particular population or a particular setting. I would say that the representative sample is really what you want to have for questions about surveillance. At baseline, what is the frequency of these different particular genotypes that are circulating in my state? Or what is the frequency rise of these different genotypes in different parts of the state? Any of those types of questions that ask about baseline incidence, changes in incidence, all of that is best approached through representative sampling. Representative sampling is hard. So with representative sampling, what you are trying to do is you are trying to capture the full extent of pathogen genomic diversity that is circulating in your jurisdiction of interest. And you are trying to capture that diversity proportionally to the actual burden of disease. So if we think about this just in terms of variants, maybe you have 5 variants circulating in your state. In an ideal representative sample, you observe all 5 of those variants, and the number of sequences from each variant that you have is reflective of the burden of disease caused by each of those variants. Representative sampling can be heavily influenced or impacted by oversampling of outbreaks or undersampling particular areas that are hard to reach. For example, in more rural areas of Washington, we have a much harder time getting representative sampling or just getting any sampling. And our urban areas tend to be oversampled, even though their burden of diseases is often higher too. So that can be really tricky to get right. I think that one of the things that really helps with this is annotation of sequence data. So for those who've heard about baseline tagging during the SARS-CoV-2 peak pandemic period, this was a way of saying whether samples were enriched because they'd been collected all part of the same outbreak, in which case you might be sort of over-representing a particular variant because you're doing intensive sampling of that variant. And you want to be able to remove some of that oversampling. And the easiest way to do that is if you have data that are tagged as being collected representatively or not. I think really SARS-CoV-2 is the only example where I've seen really systematic and widely adopted use of baseline tagging and clean design of representative sampling mechanisms. The vast majority of the time, what we actually have is a convenience sample, which is just like whatever data were available to us, whatever samples happened to be saved, whatever samples just happened to not degrade and were therefore available to have sequence data generated off of them. And that's not ideal, but convenience sampling can be rescued, and the robustness of the inferences from your convenience sample can be tested. And so you'll see that a lot of times when we have a convenience sample, you'll see the phylogenetic tree or you'll see the inferences, and then you'll see authors on that study try to replicate those inferences or the phylogenetic tree dynamics that they see on a subsample of the data. So they'll downsample the convenience sample in different ways to basically artificially try and change the sampling frame and see how sampling frame affects the robustness of the findings or the dynamics that you find. And so, the vast majority of our data and our studies in genomic epi make use of convenient samples. You can learn a lot from them, and you can have sensitivity testing of the inferences that you make off these. And so I think that can really be rescued. The last sort of sampling dynamic that I see used quite a bit is intensive samples or intensive sampling. And really, this is useful when you have an outbreak or a population that you are really interested in exploring questions about that are very specific to that outbreak, that setting, that population. So here I'm showing a bunch of sequences that were generated from a really major skilled nursing facility outbreak that occurred. This was while I was working in California. This is in Humboldt County. They had a super large outbreak in one of their skilled nursing facilities with a 100% attack rate within the resident population. And there were a lot of questions about what could have facilitated that outbreak? How did that outbreak start? Was there anything, sort of particularly worrying about the viral variant that circulated in that skilled nursing facility? Why was the attack rate so high? And so we tried to sample as many cases, or we tried to sequence as many of these infected individuals as possible. That really over enriches for whatever variant is circulating in that population, but it gives you your best bet at starting to look at epidemiologic linkage or possibly directionality or really understanding that outbreak. Importantly, you'll always need to contextualize your intensive samples with representative samples. So if you want to understand how this outbreak started, you are almost certainly going to need to take your outbreak samples, and instead of just having a tree that only brings together your outbreak samples, have those outbreak samples run alongside a bunch of surveillance sequences as well. And that will help you improve your estimates on the timing of when that outbreak began, and will also help you to understand where that outbreak potentially was seeded from. And so, while there are these three bins, really you're never just going to pick one. And in an ideal world, you always have both representative sampling and intensive sampling available to you, and you really just choose to use them based off of the question of interest. All right, so I wanted to dive a little bit more into designing representative sampling because this can be a little bit tricky to do and requires not really math as much as forethought and a real understanding of your jurisdictions and the communities that you support. And I think that that's a really important thing that the applied epidemiologists can bring to this work that often academics maybe aren't as familiar with because they don't work with those communities in the same way that we do in public health. So I mentioned in the previous slide that when we're trying to design representative sampling, that really what we want to do is capture the full extent of genomic diversity that is circulating in the population, and that we are trying to sample those different pockets of diversity proportionally to the actual frequency or sort of associated burden of disease that is causing them. So how I'm choosing to show this visually here is that blue dots are people, and colored lines are representing a particular genomic variant of this pathogen that are circulating throughout the population. And this red variant is genetically divergent from this yellow variant. So if I have a representative sample, what I want to make sure is that when I go to sample infected individuals, I capture both the yellow variant and the red variant, and that I capture both in proportion to the amount of disease they're causing, which is very similar. If you have this really nice theoretical population, where I've shown here, where every individual has an equal and identical probability of interacting with every other individual in the population, then any infected individual has the same probability of transmitting on to any other person in the population. And in that idealized version, you could just sample infected people at random, and you would get, the sequences that you get would be representative of the underlying dynamics. That would be nice. That is not the world that we live in. Instead, the world that we live in has populations of individuals that are typically quite structured. So, these can be geographic structures. For us in Washington, we have a mountain range that splits our state. And we notice that what tends to circulate on the western side of the mountains often tends to be fairly distinct from what circulates on the eastern side of the mountains, at least, initially, certainly. This is also just the typical social things that you would expect, like if there are people who tend to speak the same language or people of the same age group or anything like that, that influence them to be more likely to commingle with each other than they do with other people, then that changes the probability that they're more likely to infect someone or transmit their infection onto someone that they're more likely to commingle with. And they're less likely to transmit an infection onto someone that they're less likely to commingle with. And what that means then is that when you have circulating variation that's sort of within a pocket of the population, it's more likely to continue to circulate in that pocket rather than bopping around between pockets. So if you want to capture both the red and the yellow variants of this theoretical pathogen that we have, you need to make sure that you have sampling set up to collect samples from both or all of these different pockets of population. There is no amount of math or theory that will sort of help you with this design. This is really about knowing your populations and what impacts how they interact with each other, right? This is us in Washington knowing that that mountain range is sort of a pain to traverse. And so people don't traverse it all the time. Or it's knowing that you have two communities that are highly connected, or it's knowing that people from the north side of this city tend to very rarely go to the south side, or anything like that. And then ensuring that your sampling is set up to have or to give you visibility into those different pockets of population. So that is sort of the key behind designing representative sampling. I wanted to talk as well a little bit about the way in which different challenges with sampling can impact the inferences that you are able to make or the types of dynamics that will show up in the tree. And this is important because typically, like anything, you never get as perfect a study design or as perfect a sample as you'd like to have. And so it can be helpful to think about the ways in which sampling can shape the inferences that you get, and you can keep that in the back of your mind as a way to sort of flag yourself up. I wonder if this is the true dynamic or if I'm just seeing something that is artificial and introduced because of the sampling that I had access to. So, sorry. So one of the ways that you will frequently see this pop, I would say, is that sampling incompleteness won't make an inference wrong, but it will make the inference incomplete as well. And so I'm Canadian, so I like to use Canada as this sort of object example. Imagine here that I have an epidemic in Canada, and it's moving from these different provinces from east to west. So it originates in Ontario, moves to Manitoba, then over to Saskatchewan and into Alberta. So the true dynamic is to move province by province east to west. If for some reason I have good genomic surveillance programs in Alberta and in Ontario, but I don't have any genomic surveillance available to me in Saskatchewan and Manitoba, then I'm going to build a tree that only has genomes from Ontario and Alberta. And all of this sort of genetic diversity that has accrued during this path through these other provinces will make Albertan genomes look more diverged from Ontario genomes. And if I build a tree with these data, I will get an inference that Albertan genomes appear to be descended from Ontario genomes, indicating sort of the directionality associated with this outbreak. But because I don't have any sequence data from Saskatchewan and Manitoba, I won't see any of this transmission through those provinces. So I won't get the wrong answer per se, in that it is true that the outbreak starts in Ontario and eventually ends up in Alberta, but I will have an incomplete picture of what is happening. And this is why we're often fairly cautious about talking about directionality in genomic epidemiologic studies, especially right now when we don't necessarily have perfect sampling design. We're pretty confident that oftentimes we're not surveilling particular populations as effectively as we could be. And so we're really never quite sure what we're missing in our pictures. But the inferences aren't wrong. They're just incomplete. That doesn't mean that inferences can't be wrong, though. So where you see this pop up the most is actually with phylogeographic analyses. This is a method of phylogenomic analysis that allows you to reconstruct sort of probable states, not just at the tips where you know a particular data field, but at the internal nodes that represent ancestral pathogens that you believe circulated at a particular point in time, given the patterns of diversity that you see at the tips, but that were not actually sampled. So with phylogeographic inference, we are trying to reconstruct trait states. In this case, the picture here that I'm showing is actually host. I'm trying to reconstruct whether these ancestral MERS virus infections likely circulated in humans or in camels. And because humans tend to be particularly interested in human outbreaks and are not as good about effectively sampling the pathogen populations that circulate in animal reservoirs, we have a lot of sequence data from human outbreaks of MERS coronavirus and a lot less sequence data available from camels. And so if you use one of the most common models in phylogeographic inference, that treats the number of samples as informative about the amount of disease in that particular species, you end up getting this picture of MERS coronavirus interspecies transmission. You get it completely backwards. So you can see here that these red arrows indicate human to camel transmission, and the green arrows camel to human. So we think at baseline that probably MERS is largely circulating in camels, and then it spills over sort of repeatedly into human populations. This tree is showing the exact opposite of that. They're showing that the vast majority of the tree of MERS coronavirus diversity seems to circulate in human beings, and that then you have all of these instances of human to camel transmission. And that is what is giving you the picture of what's circulating in camels. That is wrong. I mean, that doesn't fit our understanding of how these zoonotic spillover events tend to occur. And if you use a method that actually accounts for the issue that you have here of greatly oversampled human MERS coronavirus cases and greatly undersampled camel MERS coronavirus cases, you end up getting back your sort of expectation as to what this zoonotic and emergent disease cycle would look like, whereby you can see here, even in this tree, the camel sequences in red are incredibly diverse. They are all over the tree, whereas the samples in the human populations tend to be very, very genetically similar. And that tells you something about the underlying pathogen population size. More diversity is associated with a larger pathogen population size, which is associated with higher degree of disease burden. And so when you actually account for that and you correct for the sampling bias, we get this picture here on the right And these are methods that we have. We can use this, we can do this, that shows that the majority of the genetic diversity of MERS coronavirus that we see circulates in camels. And then we have these sort of sporadic spillover events into human populations with fairly rampant, or not rampant, but fairly closely related spread within those human populations that then just sort of dies out. So this is by far and away the most egregious case that I've ever observed of how sampling bias can affect these inferences. I would say typically phylogeography is a little bit more robust to sampling bias than this example suggests. But it can be useful to think about this. And I think this can be instructive in terms of thinking about why we want, say, One Health genomic surveillance studies, whereby we really do characterize what is circulating in our animal populations as well. All right, so before we go into the last exercise together, I wanted to share what I think are the most important take-home thoughts as you start to think about study design for genomic epi. The first is, to the extent that you can, try and start here in the whirling vortex with the question that you want to answer. The second set of take-home thoughts is really around the data that you can get and the methods that you can use. So this question of how many samples you need to collect is hard to solve, and it is dependent on the question of interest up here, and also on the disease dynamics at play. It's going to be hard to get absolutely perfectly right. You probably won't get it absolutely perfectly right all the time, and we can still learn tons without getting it absolutely perfectly right. So think about it, but don't worry about it necessarily to the extent that you don't ever move forward. Secondly, representative sampling and intensive sampling are really important, and they go hand in hand. You need both. The vast majority of sequencing that is conducted by public health agencies right now, I would define as intensive sampling. We tend to only sample outbreaks for various things. That is not ideal for genomic surveillance because we end up characterizing something about the outbreak and we have no understanding about how that outbreak fits into into a larger picture of the epidemiology of this disease in our communities. And that hampers the work that we're able to do. I have my dogs with me and they're getting excited. Apologies. The last thing is that oftentimes you will have access to convenient samples of data. And while perhaps that's not ideal, they can be rescued with down sampling. And so don't worry too much about that. Just make sure that you're doing sensitivity analyses to assess the ways in which sampling frame might shape the inferences that you have. All right, so with that, I want to take the last 15 minutes to work together because I think, you can hear something all you want, but I think it starts to help if you get to think about those questions in a real setting. And so I wanted to walk everyone through actual outbreak that happened. This was a mumps virus outbreak that occurred in Washington State in 2016 to 2017. And I'm showing here the epi curve. And the epi curves are colored according to the sort of demographic group that was largely affected. So we have a significant Marshallese community within Washington State, similar to Arkansas. But during this outbreak, we saw both Marshallese cases and non-Marshallese cases occurring, and you can see that the epi curve is colored according to that status. So this is now a question for everyone on the call. I would love it if you want to unmute and share your thoughts. That would be great. But if you're not comfortable doing that, you can definitely throw things in the chat as well. I'd like you to kind of throw out some ideas. What question do we want to answer here? Or at least if you had this outbreak in Virginia, and you had, this is close to 900 cases, and they're split between Marshallese and non-Marshallese communities. What would you want to know? What would be your priority to investigate?
I think one question to ask might be if this is one outbreak that's going on between the two separate communities, or um, if there are separate outbreaks occurring at the same time.
Yeah, absolutely. That was 100% one of the things that we were wondering about. Are these just by chance two separate outbreaks that are occurring concurrently? Is transmission moving between these populations readily, or is sort of the outbreak getting started in these different communities and then just going on without sort of bopping back and forth? I think the other thing that we were really interested in is sort of transmission intensity and differential transmission dynamics that might be occurring in these different populations. So the actual number of cases of mumps virus that occurred as part of this outbreak was relatively even between the Marshallese and the non-Marshallese communities in Washington state. However, non-Marshallese people are a tiny proportion of individuals in the state. So despite the fact that the absolute numbers of cases were similar between these two populations, Marshallese individuals were in fact highly, highly enriched in the population of cases. So really we went into this with this question of how are the Marshallese and non-Marshallese outbreaks related? And are the transmission dynamics that characterize the epidemiology in these two different populations different as well? All right, so moving into the next sort of box of the whirling vortex, we want to think about the data that we have access to and how we want to approach sampling and the methods for doing the analysis. And so I have 3 questions here with hints that would guide how you could approach sampling. So the first one is, what kind of sampling frame do you want to use? Do you want to use representative sampling, convenience sampling, or intensive sampling? And nobody needs to feel shy about this, right? There's not honestly really right answers here, but I think that the practice of engaging in these questions is really like the way in which it starts to become easier. And so don't feel like there really is no right answer here. The goal is to think about your selection and what sort of evidence you have for that opinion. But you could give multiple answers here and discuss why they're correct. So no one needs to feel shy.
Yeah, I would say that I would go for an intensive sampling approach.
Cool. Can you share with me why and what population are you intensively going to sample?
Yeah, because it seems like it's a fairly contained outbreak, at least I hope, rather than something that's more endemic. I'd want as much information as I can so that I can kind of pull that into a phylogeographic analysis. I would maybe start to target the Marshallese communities and then maybe start there and then work my way out geographically to sample cases that are more closely linked in physical space. Just because if I am working on like a phylogeographic approach, that might be the easiest approach to start with.
So, thank you so much for sharing. Anyone else want to weigh on what sampling frame they might use and why? You agree? Would you do something different?
I think it it might require more of a representative sample just with the number of confirmed cases for this particular outbreak. Close to 900 cases might be pushing the laboratory throughput a little bit, depending on kind of the time frame in which the cases are being detected.
Awesome. Yeah,
This is
thank you both. Oh, yeah, go for it.
Sorry, this is Jenny. I would say I would take a more pragmatic approach, thinking about like what can we get, and I would think about what sort of I guess treatment center type locations where we could obtain these samples. So I would investigate whether or not we had like outpatient providers or treatment facilities that were perhaps common to both the Marshallese and non-Marshallese. And so that may not theoretically be possible in terms of like geographic location, but I would try to find, ways to obtain samples from both in a representative way to try to, make sure we had the samples and to get those forwarded.
Interesting. Okay. Yeah. At the time I hadn't even thought about that. But yeah, that's a cool approach. Are there any factors, before I dive into like finish things off with what we actually did, are there any other factors beyond this sort of like Marshallese or non-Marshallese status that you'd want to think about that might structure the viral population? And I have this hint here about vaccination, right? And I think to clarify that hint a little bit, people who are vaccinated and have a protective vaccination history, if they're infected, they're more likely oftentimes, or at least theoretically, to be infected with a pathogen that has somehow escaped from that vaccine-derived immunity. But that selection pressure isn't present in individuals who are unvaccinated. And so would you have any concerns about not taking into account vaccination here? About, I guess I'm not saying this well, but do you want to have sequences from folks who are vaccinated and unvaccinated, just from vaccinated, just from unvaccinated, and why?
Like, I would say it depends. I mean, I want to know from you, did you have suspicions of vaccine breakthrough, or, were these cases occurring in mostly unvaccinated Marshallese and non-Marshallese populations? But if you can't give us that hint, I think you would want, again, ideally, a more representative understanding of what's circulating by trying to sample both unvaccinated and vaccinated. That would be my thought.
Yeah, yes. So I agree. And I didn't perhaps give you the most critical part of the information associated with question 3 here, which is that Marshallese individuals are actually more likely to be fully vaccinated against mumps than the broader Washington population. And so likely the sort of larger outbreak within the Marshallese communities is not associated with under vaccination because they're more vaccinated. It also was skewed to a pretty young age distribution within the Marshallese community as well. And so we didn't really think that there was waning immunity, which is a sort of more common thing that we see with the outbreaks that we've observed on college campuses or in military barracks and that kind of thing. And so we were worried that maybe like there's not a ton of evidence for this, but we were wondering whether there might be a viral variant that was circulating that was somehow escaping vaccine-derived immunity. That was something we were wondering. And so what we wanted to ensure we collected were samples from both vaccinated, fully vaccinated, partially vaccinated, and fully unvaccinated individuals. And we wanted to see whether the genotypes circulating in people with different vaccination statuses were similar or different. Likely if you have a particular viral variant that is circulating amongst both vaccinated and unvaccinated people, then the ability of that variant to circulate is not associated with vaccination status. And so it's, I think that's really the critical thing. But this, I think, is a critical point. When we started to see vaccine breakthrough cases for COVID, a lot of people wanted to sequence them to see whether they were somehow different. And if you sequence those vaccine breakthrough cases without sequencing non-breakthrough cases, you essentially have a case control study without any controls, because it's very possible that the vaccine breakthrough cases are simply infected with whatever is circulating in the broader community. But if you don't have that broader community picture, you're not going to know that. And so that's like a common one to encounter, I would say. In terms of what we actually did, so we tried to get a really sort of good temporal coverage across this entire outbreak. And so you can see here that we're trying to so we're trying to include and generate sequence data proportionally to burden of disease. So we're trying to have a little bit more sequencing from this period of peak transmission, and we have a little bit less sequencing from when transmission starts to dwindle. And we actually tried to sample proportionally Marshallese versus non-Marshallese individuals according to their disease burden, not their population frequency. So our data set was almost split between infections among Marshallese individuals and infections among non-Marshallese individuals. And I'll show you this here is sort of the picture on, this is the phylogenetic imprints with that curve that I'm showing after the fact. And really what we wanted to see then is whether introductions are separate outbreaks within the Marshallese and non-Marshallese communities, and how cases between Marshallese and non-Marshallese communities are related. So I realize that we're basically at time. So the major takeaway here is that there are a bunch of introductions that die out really quickly. They don't yield much onward transmission. And that happens almost exclusively when that introduction occurs into non-Marshallese communities. When introductions occur into Marshallese communities, we see a lot more genetic diversity, a lot more onward transmission, indicating sort of a higher force of infection and a more rampant outbreak. We do see community samples that cluster within these clades that circulate predominantly in the Marshallese communities, but they tend to be basal within these clades, which I'm kind of showing diagrammatically up here, whereby even when you have circulation in the Marshallese community, once it sort of circulates into a non-Marshallese individual, that tends to end the transmission chain. And so while like half of these cases are in non-Marshallese communities, the vast majority of the transmission that matters and is really rampant that you would want to like most aggressively deal with is actually occurring within Marshallese communities. And that's not a result of under-vaccination. And so this really kind of changed our picture of mumps epidemiology. So I know that's a very abrupt way to end. I'm happy to stay on a little bit later if folks have questions, but I realize that a lot of people have like back-to-back meetings these days. So if you need to hop, please feel free to, but otherwise happy to stay on the line a little bit longer if you'd like.
Great. Thanks so much, Allie, for presenting today. This is really great. I also wanted to mention too that Allie has really spent time putting together other training resources as well, including her Applied Genomic Epi Handbook. And I think it's a really great resource. So I'm going to go ahead and share the link for that in the chat if others want to look at that and learn a little bit more about some of the stuff Allie's presented today.
Thanks, Alexandra. Yeah, that was a fun one to write.
Does anyone have any questions or anything? I'm not seeing any questions in the chat. So thanks again, Allie. I really appreciate you talking to us about this.
Sure thing. Thanks for having me. Great. Talk to you soon.
Thanks. Bye.